Good afternoon. I'm Dr. Mark Israel, the National Executive Director of the Israel Cancer Research Fund. I'm delighted to invite you to the fourth in our webinar series, Brilliant Minds. In this series, we interview various Israeli cancer researchers whose research is funded in part by the Israel Cancer Research Fund. Today, we're going to introduce and interview Dr. Yossi Shiloh from Tel Aviv University. Dr. Shiloh works on DNA repair and is truly one of the international leaders in that field. DNA repair, of course, is important for radiation therapy because the way in which radiation therapy, a mainstay of cancer treatment, works is by damaging the DNA in tumor cells. And of course, if the DNA is repaired, tumor cell goes on to live and the therapy fails. And so understanding the basis for DNA repair is a key aspect of understanding how to deliver radiation therapy optimally. And today I'm so pleased that we have with us a strong supporter of Israeli cancer research, a member of the New York City chapter board of the Israel Cancer Research Fund, a past gala chair, and most importantly, a cancer survivor of breast cancer, Mrs. Andrea Greene, who received radiation therapy as part of her therapy. Andrea, I'm so pleased to have you with us this afternoon.
Hi, Mark. It's good to be here, and it's good to be seen.
Well, it's just grand to have you. And Andrea, a key part of cancer treatment, one of the central modalities amongst chemotherapy and surgery and radiation therapy, and of course now we have immunotherapy as well, but is radiation therapy. And I know as part of your treatment for breast cancer, thank God, long ago, you experienced radiation treatment, and we're going to be talking about some of the science behind radiation treatment going forward with Dr. Shiloh. But could you give us a little idea of how radiation fit in with your treatment? Was it the first treatment you got, or how did it go?
Sure, absolutely, Mark. We started with mid-August. I was diagnosed, and within maybe 36 hours, I was in surgery. Had a very successful lumpectomy and sentinel node biopsies. And fortunately, while the cells left the site, they did not travel far enough for me to need anything more extensive than the lumpectomy. So that was a blessed thing. After that, we waited 3 weeks and I started chemotherapy. I had adriamycin Cytoxan, and I had that for the next 12 weeks with my oncologist. And that was tough. I'm not going to pretend it wasn't. That was really very hard. In between chemo and radiation, I did my genetic testing because it was very important to me to know whether I had BRCA1 or 2, which were the only 2 genes identifiable years ago. Today, of course, there are many more genes. So after I received the report that I did not have anything genetic that they were able to find, I proceeded with the radiation treatment, and that was mid-December, and I did that through the beginning of February.
Mm-hmm. Now, typically when breast cancer patients receive radiation therapy, they receive it over some number of weeks, and they go repeatedly each week for a radiation treatment. How was your radiation administered?
I went 5 days a week, very, very consecutive. We tried very hard not to miss a day. It was very important that I stay healthy because they had expressed that if I was to get a cold or run a slight fever, so they checked my fever, my temperature on a daily basis. The last 6 days of my radiation was a more targeted therapy, which they at that time called a boost. So after I believe it was 30 days of treatment, I then had 6 days of boost for a total of 36 days.
Well, that's quite an experience. Now, one of the things that most cancer patients do fear are the side effects of treatment. And I'm just wondering whether or not you experienced what you identified and your doctor identified as side effects of radiation therapy that might be informative for our audience.
I think overall I had an extremely positive experience with radiation therapy. Really I was never sick. I never had any burns. I was never tired from it. The only symptom I'm going to say that I had was that I felt hot a lot. And sleeping at night was difficult because I felt hot, and it was the middle of winter. My husband was wearing sweatshirts and sweatpants to bed, and I was running the air conditioner. So luckily, my radiologist had suggested a medication that she thought would help with that heat sensation, and it did. It was terrific, and I'm going to say that for the most part, radiation therapy was the easiest part of my cancer treatment.
Well, I'm sure our listeners will be glad to hear that. Just before we wrap up this part of the webinar, I'm wondering if you have any suggestions or thoughts for patients who might be anticipating the start of radiation therapy for a cancer diagnosis or are actually getting radiation therapy now.
Well, I think you have to use good judgment, like with anything, and take good care of yourself. I didn't bathe excessively because it would have been an irritant. I didn't put lotions or perfumes or deodorant on during radiation. If I was not comfortable on the radiation board, I was in a prone position. So I was face down on my stomach, 6 feet up in the air. And that's how I had radiation treatment. But if for any reason I was not comfortable with the position, you have to speak up. You have to say, this is not comfortable for me, because you don't want to walk away with a pulled muscle or a stiff neck or, something that makes you more uncomfortable. And everybody is extremely accommodating. Don't hesitate to say to your doctor if you're having any symptoms or any side effects. They have an answer for every one of your questions. They truly do. So speak up, be your best advocate.
Andrea, I so appreciate your candor and thoughtfulness and your participation today. Thank you very much.
You're welcome.
And now I want to turn the focus of our webinar to an interview with Dr. Yossi Shiloh. Dr. Shiloh is a professor of human molecular genetics at the Sackler School of Medicine at Tel Aviv University, and he serves there for some time now as the chair of the Department of Human Genetics and Molecular Medicine. He's going to be interviewed by my friend and colleague and the chair of ICRF's Scientific Advisory Council, Dr. Nancy Maizels.
So thank you, Mark. Thank you for being here, Yossi. I'm Nancy Maizels. And I would especially like to thank Andrea for sharing her story, and especially for reminding us and encouraging us how important it is for anyone confronting a difficult diagnosis to be empowered to be one's own spokesperson. We heard that from Linnea Olson in a webinar about a month ago, and it's a really important thing to hear. Stories like Andrea's inspire all of us to work harder in the lab and in the clinic, and in ensuring that there's funding for excellent cancer research. And ICRF has been working to do that since its founding in 1972. Its mission is to fund the very, very best cancer research at the top universities and institutions throughout Israel. And ICRF has funded 2,500 grants with a total of over $72 million in almost the last 50 years. And currently we're funding 72 different investigators with a budget of about $5 million a year. We began these webinars only in the spring when we realized, partly because of the pandemic, that there was a large and enthusiastic audience who were interested in learning more about human health and disease. And so we've begun the webinars, and we're glad to see how many people there are who care about this. The webinars, like ICRF's mission, depends upon hard work and funds. And we want to reiterate our thanks to Andrea and to her family for their enormous generosity, for Andrea's work as a board member, and their very strong and continued support of ICRF. So the ICRF funds a professorship, which we're delighted that Yossi Shiloh holds. And I'm about to have the pleasure of introducing Yossi, but before, I'd like to reiterate the context that Mark told you about that will make you understand why Yossi's research has been so important to cancer. An interesting fact that I keep being surprised by is that even now, about half of all tumors are treated with radiation. It's the single most common treatment, even as we see more and more fabulous specialized treatments being developed. The mechanism by which radiation works is it makes DNA breaks, destroying the integrity of our genomes. And that especially matters to cancer cells because they're replicating very fast and they don't take the time to repair bad accidents. Normal cells, in contrast, are very careful about repairing DNA breaks. So radiation inherently is able to attack cancer rather than normal cells, which is the real trick for a good cancer mechanism, and to do damage that prevents them from proliferating. So today, Yossi's going to tell you about his discovery of a gene nicknamed ATM, which is a control center that tells the cell how to maintain the integrity of genomic DNA in the face of stress like radiation. There's a longer story that is on the website, an interview with EMBO Reports that Yossi gave about a year ago. That's absolutely fascinating. I encourage everyone to read that. Briefly, I'll sum up that before Yossi achieved all these things, he was an undergraduate at the Technion, a graduate student at Hebrew University of Jerusalem. He did a brief postdoc at Harvard Medical School and came back to Tel Aviv University, where he's now a professor of human molecular genetics at the Sackler School of Medicine and holds an ICRF professorship. So welcome, Yossi. We're so delighted that you've agreed to talk to us today. And I'd like to begin by asking you, how did you become interested in science?
Well, first of all, let me thank you for the invitation and Nancy for the introduction. And I'd like to join you in wishing Andrea and her family excellent health and many, many more healthy and happy years and of many contributions to science, to cancer research, everything. And the same for everybody who's in the audience right now. So to go back to your question, I guess that started when I was a high school student. And actually, it's quite an interesting story because I think I owe my interest in science to my chemistry teacher. Her name is Dr. Bracha Orgad. She immigrated to Israel from Argentina. And believe it or not, I'm still in touch with Dr. Orgad. She's in her 90s now. Just 2 weeks ago, we spoke on the phone. She's fine. And her chemistry lessons actually were, I think, the first that made me interested in life sciences. And then I became more interested in biology. And when I studied at the Technion, chemistry and biology, I had a wonderful genetics course. And I decided that genetics would be my field. And since I was interested in medicine too, I chose a field in genetics, which is intimately associated with medicine, and that is human genetics, medical genetics. And I went to Jerusalem to do my master's with the late Professor Maimon Cohen, who had just established the first department of human genetics in Israel at the Hadassah Medical Center at that time. And I think I was his first graduate student, and I did my master's with him, actually on the chromosomal abnormalities in bone marrows of patients with leukemia and pre-leukemic states. That was my master thesis. Still not directly associated with radiation biology and all that, but then the story goes on.
So tell us the story.
Okay, so for that, I think I would like to share my screen with you. Can you see my screen now? Let me share
Yes,
it.
perfect.
Okay, wonderful, great. So I'd like to start by showing you Maimon's photo. He was a wonderful scientist and a wonderful human being. And I had very enjoyable period of studies with him when I was a master's student, but then I had to move on to do my PhD. And Maimon knew I was looking for a good subject for my PhD thesis. And he came to me one day, it was in the summer of 1977, and offered to me to join him on the following day on a field trip. And a field trip that geneticists do is basically visiting a family with usually a family with a certain genetic disorder, collecting blood samples, drawing the family pedigree, and giving the family genetic counseling. And we were going to see a Moroccan Jewish family living in a small village in the southern part of Israel, actually the Negev desert. They had 10 children, 4 of whom were affected with a very rare genetic disorder, with a long name and strange name, ataxia telangiectasia, or AT for short. I had never heard about AT before, so I quickly read about it. And here you can see the general, quite amazing clinical picture of this devastating disease. As you could see from this image, it affects the nervous system. So there is neurodegeneration affecting specifically this part of the brain, which is responsible for coordination of movements, and lack of coordination of movements is called ataxia. So what is telangiectasia? Telangiectasia, oddly enough, is widening of blood vessels in the eyes, which gives the eyes the appearance of like bloodshot eyes. Very strange combination, but it's not only that. There is immune deficiency in this disease on top of the neurodegeneration, and the patients are extremely prone to respiratory infections. By the way, these days, you can imagine that they have to be kept really, really well and isolated so they won't catch COVID-19. And if all that is not enough, they are cancer prone. They have a tendency of about 1,000-fold higher compared to the general population for developing leukemias and lymphomas. And they are also sterile. And then, and this is really kind of cruel irony, as if this whole combination was not cruel enough, they're extremely radiosensitive. Now, what does this mean in real life? This sort of neutral word, radiosensitivity. What it means is that a long time ago, when AT patients were treated for the first time with radiation, the main theme of our meeting today, believe it or not, they died because of the treatment. The treatment that was supposed to save their lives actually killed them within several days. That's what it means, this radiosensitivity. AT is still the human genetic defect that gives rise to the most profound sensitivity to ionizing radiation. That's the kind of radiation which is used in radiotherapy, which is known in humans. So all this obviously made me very curious to see the family and meet the patients. And we met them in this house. And here is a diagram of the family, the pedigree. These are the parents. These are the children. Some of them have the families of their own, some of them healthy. The affected ones, those that have AT, are black solid figures, these three girls and the boy. Actually, this boy and girl were the first 80 patients that I met on that day. And please note that the elder daughter, who is healthy, she married her cousin, and they have two children with AT of their own. So second generation of the same cruel disease in the same family. And so we interviewed the parents and we saw the patients, and I could almost say the rest is history, because I decided there and then that I would do my PhD thesis on AT. This picture was taken many years later. This is actually a fun day organized by the Israeli AT Society, which was established by parents of AT children for the welfare of these kids and in order to support the families. And what you can see here, that in addition to North African Jews, as far as the Israeli population is concerned, AT affects Arab, Bedouin, and Druze families. And in fact, AT was found throughout the world in all countries. Still a rare disease. And it's one of the 6,000 or so rare human genetic disorders. I'd like to go back to the radiosensitivity, which is not only the main theme of our meeting today, but that was actually what I was studying when I was a grad student. I grew up cells, you know, that we can grow cells in culture from people. Usually the origin of such cells are either blood samples or skin biopsies. And indeed, when we irradiated cells taken or grown from healthy and unrelated individuals and cells from patients with AT, we could see in the culture dish a clear reflection of this enormous radiosensitivity that killed those first patients, AT patients who were treated with radiotherapy. Because at very low doses of radiation, that cells from healthy individuals can suffer and can grow after that without any problem. They can tolerate them. Those very low doses killed the entire cell population when the cell lines were derived from AT patients. So that was a very dramatic demonstration, of course, without touching the patients. We did all the experiments in the lab, and we knew that this profound radiosensitivity was probably the key feature of this disease in terms of understanding its physiological and molecular basis. Certainly, this is not the most dramatic symptom. The neurodegeneration is the symptom that makes AT so devastating. And now, these days, we don't irradiate AT kids. And so the radiosensitivity is not so meaningful to them. But to us, it meant a lot. And what it meant to us was that the cells from these patients with this disease, probably have a problem responding to DNA damage that is caused to the DNA by the radiation. So let's look at DNA damage. You all know that our precious, valuable DNA is also a very delicate molecule, and it can be damaged by a whole variety of environmental agents, including some that are included sometimes in our food. But you know something? Everybody talks about radiation and chemicals, but in fact, most of the damage that is caused on a regular basis, on a daily basis, as we speak to our DNA, is in fact caused by what we call the enemy from within. These are byproducts of our normal metabolism that are produced in the course of metabolism. We call them reactive oxygen species. But these molecules that are normal byproducts of metabolism, they can damage the DNA very, very seriously. So in fact, our DNA is under constant attack, even when we do not ingest food with various bad chemicals or inhale bad air and so forth. And of course, DNA damage can lead to changes in DNA structure or sequence, cause gene mutations, which in turn can lead to cancer or simply kill the cells. So how do our cells deal with this constant attack on the integrity and the stability of our DNA? This cartoon actually represents the DNA repair mechanisms, and these tireless workers that actually keep repairing the damage, responding to damage all the time, are proteins whose primary mission is to do just that. And you can realize by now from what I've just told you that they have an ongoing duty and are working all the time in order to repair the damage that is caused in the DNA all the time. In fact, tens of thousands of lesions per day in every cell. And they are quickly repaired because these DNA repair mechanisms, which are very complex, and they deal with all the various DNA lesions caused by these chemicals and radiations, and they repair them very, very quickly.
So
But
Yossi,
now,
to be sure we'll have time for questions from the audience in a few minutes, I want to be sure you get a chance to tell us about the work you're currently carrying out.
yes,
As a
so
professor, I'm sure you're headed there.
yes, so basically what happens when some of the genes that encode these proteins is defective, you can imagine that when these tireless workers are not there, a very serious genetic disorder is caused. And we call these disorders genome instability disorders because, like AT, they lead to extreme sensitivity to DNA damaging agents and degeneration of tissues and so forth. So we're going back to ionizing radiation. So the question was, what is that DNA lesion that AT cells cannot repair well? Indeed, it is DNA breaks, simply tears in the DNA. This is a very, very serious DNA lesion. One unrepaired break can kill a cell or make it cancerous. And the system that repairs double-strand breaks, respond to them, is extremely complex. And we found, and other labs found the same, that in AT, the problem is profound defect in the cellular response to DNA breaks. Okay, so what's next? Next thing was to identify the responsible gene and see what the protein product of this gene does in order to deal with these breaks. And this is what we did in our lab in Tel Aviv. And in 1995, we identified the responsible gene, the gene which is mutated in AT patients. And we dubbed this gene ATM, which stands for AT mutated, the gene which is mutated in AT patients. And we found what the protein that is encoded by this gene does. And that protein too is called ATM. Look at this amazing cartoon. What ATM in fact is, as we expected, the master controller of the cellular response to these breaks. And it does so by recruiting many proteins. All these little boxes are the proteins, and it also has the capacity to modify them and make them serve the system for a while so the breaks can be quickly repaired. This is an enormously complex system, as you can see here. And it is ATM that mobilizes the system and actually regulates it. And my favorite metaphor is that of an orchestra conductor. This is exactly what ATM does. And when ATM is not there, and this is exactly what happens in AT patients, we found that AT patients have no ATM at all. And
So that's
this is how their disease is caused.
that's a huge orchestra. There must be 1,000 different proteins.
Exactly. And believe it
Yeah.
or not, we have an even more complex diagram that shows 1,100 targets, as we call them, of DNA. And going back to the musical metaphor, it reminds me of Mahler's Eighth Symphony, the Symphony of the Thousand, which is called so because you need 1,000 musicians and singers on the
Yeah.
stage in order to perform it. That's exactly what's going on.
Yes.
Now, you might be asking yourself, so what's the story with ionizing radiation? It has kind of Janus phase. On one hand, it causes this terrible damage and it needs to be repaired. And if it's not repaired well, there is radiosensitivity and this terrible disease, and it's all bad. But on the other hand, we're talking about the very same radiation as one of the major means of cancer therapy. So what's going on here? Is ionizing radiation a friend or foe? Well, it goes back to the DNA damage, to those breaks. That's how ionizing radiation affects cell survival. And it's all a matter of how you irradiate tissues and cells, what is the radiation dose, the radiation regimen, and how you focus the radiation on the tumor. And as was already mentioned here, I think Mark mentioned that in the tumors, you have very rapid cell proliferation. The cells multiply, they divide all the time. And this is the situation where cells are most sensitive to radiation because the breaks that the radiation causes are extremely critical and need to be repaired very quickly when cells proliferate. So if you design carefully the treatment regimen, you focus the radiation beam directly to the tumor, you can achieve the goal of eradicating the tumor. Still, there might be some side effects, and unfortunately, many people are aware of it. Some nearby normal non-tumor tissues can be affected. There are sometimes side effects, but if you design the treatment regimen carefully, you can achieve the goal.
So
Now,
Yossi, this leads directly into a question that's appearing on my Q&A screen, and I'd love you to answer it now. So the question is, has radiation treatment changed over the 15 years since Andrea received the treatment she told us about? Is these the improvements that limit?
Yes, the treatment is constantly being improved. Actually, what I was going to say is exactly the answer to this timely question. Because the treatment regimens are getting better and better, not only because of the machines are better, but also because the doctors, the radiation oncologists, understand better the biology. You have, in order to really do a careful design of the treatment regimen, you have to know what the radiation you are using is doing to the cells. And that takes us back to ATM, the chief mobilizer of the cellular response. So you need to know how cells will respond to this radiation in order to design a better treatment. And let me give you some food for thought. We have now very good inhibitors of ATM. This means that these are chemicals which, if we add them to cell culture of normal cells that have normal ATM, not cells from AT patients, they stop the activity of ATM, thereby rendering these cells very sensitive. We know how sensitive cells are when they don't have ATM, like cells from AT patients. Only that in this case, these are normal cells with a chemical that keeps their ATM in check, inhibited. Now, think, for example, of the possibility of introducing an inhibitor of ATM directly into and specifically the tumor cells. Obviously, you will make the tumor much more sensitive. And what this means is that you would be able to focus radiation on tumor, but at a much lower dose, because the tumor cells will now be extremely sensitive, and you won't need the high dose that we currently use. You can go much lower doses, which will have lower side effects. So here is an example of how basic research could allow the doctors use more and more advanced means to make the treatment better and more refined. And let me just remind you where we started. It was a strange genetic disorder. And this goes back to lectures that I give a lot to the public. And the title of that lecture is always the same: Investigation of Rare Human Genetic Disorder, a Mission with New Biomedical Insights. And this is why it is so important to study such rare genetic disorders that nobody has heard about. You can get very, very profound, critically important and useful insights about our biology and medicine from studying such rare disease. And that's what I had in mind when I saw those patients in the summer of 1977.
That's an amazing story. And certainly AT was one of the genes that was, or ATM was, that turned out to be the most fruitful places to concentrate. But also your lab has been unusual in that as you studied it, you thought, well, we can identify the gene, but now we can find out what it does. And not everyone has demonstrated such a lifetime commitment to the problem of what a gene does, or found such a great gene to study. So you answered a question that's appearing in different forms from the audience, which is, what does this mean for therapies? Are there therapies that are in the clinic that take advantage of identification of the ATM gene or the genes that it regulates?
I already mentioned one example. This is not still in clinical practice, but obviously people are trying now. The inhibitors of ATM are there, and they are getting finer and finer. We use them in the lab for our studies, but it's a great problem that remains, is how to direct this little inhibitor, this small molecule, specifically into the tumor. And once this is achieved, we could, so to speak, sensitize specifically the tumor cells to the radiation using the inhibitor, and then lower the dose of radiation and treat the tumor more successfully. And again, I can't emphasize enough the importance of basic research, apparently so unrelated to the clinic, a human rare genetic disorder and all that, and see how it connects to the clinic. And indeed, I can assure you that radiation oncologists follow the ATM literature very closely.
Very
and
closely.
Believe it or not, there are now tens of thousands of papers about ATM. Every morning I find 5 new ones.
That's really
much
exciting.
To our delight.
Yes, of course. So this leads me to a question that I know many people are asking themselves, and that I as a scientist always have. Things seem to go very slowly in science most days. Occasionally there are amazing days, but most days they either go forward slowly or backwards faster than you would have wanted.
Absolutely.
And then in retrospect, things seem to go very quickly when you just date it by years. What are your insights into how that's affected your research into ATM? And then, of course, this is on the top of people's mind because of the pandemic, and I'd love to hear your thoughts on this.
Yes. And I think when I reflect back, I think that our case is quite typical. I remember the time when we were trying to identify the gene responsible for AT. We still didn't know we would call it ATM. And that was an 8 years effort. 8 years during which the entire lab was doing just that. And it was tough competition too. Many labs were trying to do the same. And the families, the 80 families whom we used to meet regularly, we still meet them regularly, they knew that the next big step in AT research would be identifying the responsible gene. And you can imagine that during those 8 long years, they kept asking us, so is this close? Is this going to happen soon enough? And I always used to tell them, look, we're doing our best. We're using the best means that science can provide us. We're trying even to contribute to developing some of them ourselves, which we did. And it can happen at any time, all of a sudden, tomorrow, a week from now, a month from now, or a year from now. And you know what? Let me tell you something. I remember vividly the day when we realized we found that gene. And we had an experimental result. We were looking at various genes. We call them candidate genes. And we knew that one of them could be the gene responsible for AT. And the way to identify the real AT gene was simple. You look at the gene sequence and structure in people unrelated to AT families and in the patients. Clearly, the gene responsible for this disease would be mutated, would be defective. Its structure would be damaged only in AT patients, but not in healthy, unrelated people. And one day, our lab manager, our dedicated lab manager, Dr. Yael Ziv, came into my office, and she was holding an experimental result in her hand. Results obtained with an Arab family living in a small village in the Galilee, in the northern part of Israel. And she said to me, and we were looking at the structure of several genes in that family, in the patients and in the parents and unrelated people. And she said to me, I remember her words, there was something going on with this gene in this family. And I looked at her result and I kind of stopped breathing. Clear that this particular gene was defective in the patients and his family. And I knew it was it, but you know, you have to be very careful. And this is something you learn over the years. So I looked at Yael's eyes, same way I'm looking now into this camera. And in the same tone I'm speaking now, I said to her, Yael, this looks interesting. Why don't you repeat this experiment, add more controls, more patients? It was a big family. There were additional patients. Come back to me with another experiment on the same family. But I knew that
You knew in your heart.
Yes. Yeah, I knew it. And that's how it happens in science, you know. You know, that same day I was giving my class, actually, gave my lecture at my students. It was 5 minutes after I came back, and Yael wanted to see me. And, you know, a few minutes earlier, I didn't know that it would be like 3 minutes from now, not tomorrow and not a week from now, not a year from now. And that was after 8 years of like a 24/7 work.
So one of the wonderful things in the EMBO Reports article, there you have lots of honors and awards, the Israel Prize, many things. But one of the things that impressed me was how you said that many labs were working on this. And instead of racing into print alone, which many investigators would have done, you got all of your competitors and colleagues together, and a single paper was published that changed the world. And that act of generosity, I think, is one of the many things that should be to your credit, in addition to the prizes and all the things that don't reflect character, but simply scientific achievement.
Quite frankly, you know, this is one of the best decisions that I made in science. You know, there were altogether 7 groups competing. And at that time, there were many such races to get disease genes, cystic fibrosis, for
Yeah.
example, and Huntington's gene. And in many cases, there was this not only competition, but outright rivalry between groups and bad feelings. And there was one winner who takes everything and leaves everybody else in the cold. And what I did soon after we found the gene was, first of all, I picked the phone and I called everybody and told them that we have the gene. I didn't want them to read about this from the paper. And in one of these telephone conversations, I called the head of the group and I told him, we have the gene, we found it, it's the gene. And there was a long silence, and then I heard him saying, I'm happy for you and I'm sorry for our group. And when I put the phone back, I knew what I would do. And I picked the phone again and called everybody. And I said, look, I want everybody to be in this paper. This should be a celebration of the entire community. And this sort of set the scene for, it was a big feast, actually. Everybody was celebrating. And this goodwill and good atmosphere has remained in the field.
Which is just amazing. So Yossi, I'm afraid our time is up. I could keep talking and listening about this all morning, but I want to thank you. It's really been an honor and a pleasure to speak with you.
It's been a pleasure to speak with you.
And maybe you'll go into the lab today and there'll be a project that's been brewing for 8 years and you'll get, what would be wonderful. You deserve it. And I'd like to thank everyone who's listened to today's webinar. I want to tell you that there'll be more webinars in September and October, and you can look at the ICRF website to find out information about those webinars. And ICRF had a gala about 2 weeks ago, hosted by Jason Alexander, who many of you may know from Seinfeld. And a friend of mine called it lightning in a bottle. And if anybody's thinking of what could they do on one of these long pandemic evenings, I encourage you to tune into that. Yossi, thank you so much again. This was just terrific.
Thank you so much, Nancy. Thanks everybody. Thank you one and all. And it's been a sheer pleasure to speak to you today.
Great. Thanks to everybody. Bye-bye. Have a great day.